Hello.
Hi,
Hi, how are you?
I'm pretty good. I'm not doing enough research, but and I went to my convocation.
Oh,
So
okay, that's great.
so I have a hat with a tassel on it that says 2025. I got a free lunch.
So you're also in the education college or something like that?
I have post-baccalaureate in education,
Oh, okay.
and I taught high school at the Adult Learning Center near here for 11 years.
Oh, okay. That's good.
So most of the time that I was attending, it was attending here. It was during class. If there were no students, I would attend online.
Okay. That's pretty good.
Yeah, a lot of it was in the learning center. Well, some people would turn up at 9 o'clock, but not very many. Like they tended to wander in at 11. Yeah. So it's a one room classroom situation, and I taught 12 subjects, but it was on an individual basis, and usually I didn't run a classroom except in the evening for people in math. I did teach physics like that once, and that worked better than them studying physics on their own. I have to say that much.
Yeah.
And I did run some biology classes too, but mostly it was math. That's what they needed. They needed just a really good grounding in math.
Oh, okay.
Yeah.
Well, that's great. Yeah. And it helped you understand more math so you could do your dissertation.
It did in a way, because I call it baby math, but also the distance ed people had me teaching calculus, and that helped me with some of my foundations in calculus that I seem to have forgotten.
Yeah.
So, yeah. So it helped me with calculus.
Yeah.
Yeah. Because the math, I taught all levels.
Yeah.
So, anyways, yeah. So I don't know. Yeah, 12 subjects is a lot. Now, overall, I think I taught 21 subjects. I taught anatomy and physiology too at the college level.
Okay.
So that really helped me.
Yeah.
When I hit biomedical engineering, they said, well, you have to take anatomy and physiology, and we want you to do it with all these other things. And that was a lot of courses for you to take all at once and try to get an A plus standing in.
Right.
So I said, I taught it, so I know it. And they believed me. So, well, I proved it. I sent them the course outline and stuff. So, and that also helped me when I was presenting my thesis.
Right.
Because they said, you said here that, proteins change conformation and also physical properties in with changing temperature and with changing pH and so on. So how can you ensure that your model is going to work all the time? And I said, in a mammalian cell, that's very tightly controlled. Period. So it was a good answer.
Yeah. Yeah.
Yeah, absolutely. Anyways, yeah, my background in anatomy and physiology has certainly helped.
Yeah.
Because this is it was a biology and mechanical engineering thesis.
Right. Yeah, it's always tough to switch between them. Yeah.
I didn't have Anyway,
Yeah.
yes.
Oh, it's always tough to switch between them. Like, a lot
Oh,
of times your committee members will be conversant in one but not the other, and then others will be conversant in one but not the other. You have to
yeah.
bridge that
Well,
gap.
the one fellow was from dentistry, so he kind of knew the mechanics,
Right.
and he definitely knew the biology. And then the other fellow was from biosystems engineering, and he knew the mechanics and some of the biology.
Yeah.
But he admitted that I knew more biology than he did.
Oh, yeah.
So, yeah. And the third fellow just said, I'm here to listen. Explain it to me so a normal person can understand this. He's not a normal person. He's very intelligent,
Right.
but he's electrical,
Oh, right.
an electrical engineer. And if there was
Yeah.
such a thing as a mathematical engineer, it would be him.
Okay.
Yeah, he's very heavily into math, the math behind optical coherence tomography. So,
Oh, okay.
yeah. So I don't know. That's an explanation of that, I guess. So yeah, and by the way, never try to teach computer science over the phone. It
Why? What?
doesn't work very well.
Just like, what do you mean by that? Like, what were you trying to do?
The distance education people had me teaching the beginner computer science over the phone, and we were programming in, was it in C?
Yeah, that's kind of tough.
It was like, okay, it was C++. That's what it was.
Yeah.
And first off, I had to find the right version of C++, buy a textbook about it so that I could get the programming right on my computer. That was a mess. Anyways, and you try to explain some things over the phone to them. Please do, like they did have a booklet where they had exercises, and they just were not understanding that learning computer science is kind of a discovery process where you do something, you make a mistake, and then you learn that's the error message and that's something you don't do.
Yeah.
And I said, you have to do the exercises in chapter 2.
Yeah.
Or you won't get anywhere. Have you done the exercises in chapter 2?
No.
Obviously they hadn't, and they're going, this is a bunch of noise. And it was like, I can't help you.
Yeah.
We're going to fail. I'm sorry.
Yeah, I mean, it's like that visual component as well. Like, if I explain to you over the phone how to program something, maybe you can get it, but you wouldn't get it. Like, you wouldn't understand how to fix it or how it works. You just don't know like every line and every command.
Yeah. And Distance Ed had an online video method at that time. Like I could have done it over the internet, but the super religious colonies out here wouldn't have it. So these boys really wanted to learn computer science, but they didn't want to do chapter 2 thoroughly.
What was chapter 2?
It was program this line, hello world. Type in hello world, get it up and running, and type this in, and then enter, and it should type hello world for you. And also, if you change the semicolon for a comma, what happens? And like just sort of general syntax rules going through them. And it's like, for some reason, they weren't doing it. I don't know what was wrong. They were at the other end of a phone. Maybe they went to class and then then went outside and played ball. I have no idea.
Yeah, it's hard to, yeah. It's like, I'll wait on the phone here for 15 minutes while you do this.
Yeah, well, the teacher tries to tell you to do chapter 2.
Yeah.
Yeah. Anyway, the phones and computer science, it's too visual for that unless you're, well, if you're blind, you can definitely learn computer programming, but you have to pay more attention to, well, the syntax of whatever you're doing,
Right.
whatever language. Anyways, it didn't work.
Yeah.
Yeah. Either they got it or they didn't get it.
Yeah.
And it was Hutterites colonies that I was teaching into. And there was one genius, and she was I guess a grade 12 student. And she was like, she said, yeah, she understood what I was talking about, but the other students, even though she got it, the other students weren't getting it.
Yeah.
So I still don't know where the disconnect was, because she was right there with them and she understood it,
Right.
and she couldn't explain it to them.
Oh, well, yeah.
So I don't know. Yep. So that's for anybody. Do not try to teach computer science over the phone.
Yeah. Looks like Lalith is here. Hello. So I did want to go over some things like we talked about, I think 2 weeks ago. Jayadratha asked about some Connectome datasets. And we have some Connectome datasets in the group. We've actually published 2 papers in the group on Connectomes. I'm going to go over that now. The first dataset is in this repository, DevoWorm Embryogenetic Connectome. And so this has a bunch of data files. I think it's GraphML files and CSV files. So these are connectomes at different stages of development, or at least the developmental cells at each stage of development. So, we have the developmental cells at their nomenclature, and then they terminally differentiate into neurons that have a different nomenclature. And so then these files talk about, first of all, the cells that are involved in the connectome at different stages of development, and then those cells as they differentiate into neurons. And so there's like an interactome, which is like the cells pre-connectome. They're interact with, the idea is that they're born, the cells divide, you have cells in the embryo. They're signaling each other. They're paracrine and juxtacrine relationships, meaning that they're exchanging chemical signals and things like that. And then eventually you get this connectivity through gap junctions. I think you have gap junctions in the developmental cells as well. But you get this connectivity through gap junctions that is verified in the connectome. So then that becomes sort of those cells when they're born, when they terminally differentiate, they get put into place in that connectome. So this all these files describe the sort of the emergent connectome at different stages of development. And so that is associated with a paper that was published in 2018. This was in BioSystems. This was the emergent connectome in C. elegans Embryogenesis. And it goes through a lot of the analysis of these data. I don't think it really shows the entire paper here. I'd have to go through and open up the PDF. But basically, this paper discusses this data set, shows some analyses, but there is source data here. Actually, maybe we have some images. Okay, so this is actually interesting. One of the things they did in this paper was to take all of the cells in the connectome and plot them out on a model of the worm. And this is from the OpenWorm browser. So the OpenWorm browser is a model of the adult worm, and you can open it up in 3D, and you can label cells based on their identity. So each cell in the adult worm, you can highlight it in the model, and you can get a description of what that cell does and its properties, but you can also kind of highlight groups of cells. And so this is showing the connectome. And so these are all the neurons in the connectome. You notice there are a lot of neurons up by the head, in the pharynx, down the side of the body, which we call seam cell associated neurons, and then we have a cluster of neurons in the tail. And the idea is that some of these have gap junction connections, some of these have axonal and synaptic connections, and then this forms the adult connectome. Now, what we've done then is just to simply say, okay, at each time point, we get a certain fraction of those cells born. So if we survey the embryo at like 400 minutes of development in the embryo, how many of those cells from the adult are born at that point? And so it's maybe, a fraction of those cells. And so you can plot those out in the adult model to show kind of where the first cells are born, what location in the adult those are going to be, and where later cells are born. And it turns out that you get, of course, neurons in the head in this area here first, and also in the tail pretty early. Not the very first neurons that are born, but you get them a little bit later on. So you get the head and the tail where you have neurons, and then of course you get these neurons along the seam cells along the lateral side later. And that seems to hold true for the worm as it develops post-embryonically, is that you get cells born along the seam, epithelial cells. And so this is, very useful, I think, for understanding this process. This is a figure. Oh, this is a table where we show kind of like at 400 minutes, there's a mapping between the adult cell nomenclature and the lineage nomenclature. So you have, say, for example, ADFL, which is a neuron, and that comes from this lineage AB, A, and then there are these L and P letters in the suffix which describe like division events. They describe things where it's, I think, left and posterior and anterior, so that they divide in different directions, and that's what the lineage means. So we can link this identity to a lineage tree, and we can link this adult identity to the OpenWorm browser. So we can do that kind of mapping between two different data sets, two different nomenclatures, and get a sense of how this works. And then, of course, we have an annotation for each cell. So ADFL is an ampid neuron, ADLL is an ampid neuron, ADFL is an ampid finger cell, AIML is a ring interneuron, and so forth. So I could look at a map in the embryo of these newly born neurons and ask, why are they born so early, and maybe find some functional significance. And because the paper is now like 7 years old, I don't remember a lot of the findings in that sense, but go read through the paper, and I believe we make some interpretation there as to some of these annotations. So that's this data set. This is all in the embryogenetic connectome repository of DevoWorm. And so this just has, a lot of graphs, a lot of data sets, and tables that are sort of in a form that basically the idea behind this repository was, of course, to have data available, but also for kind of a place for people to come after they read the paper and get access to these things and get access to some supplemental materials. Yeah, it's a very, very good paper. And then the second one here was published 2 years later. This is another paper, but we have another data set. And so this is DevoWorm raising the worm brain. So, the reason there's two separate repositories is because they belong to two separate papers or two separate entities. But they're basically, it's a follow-up to that paper. And this paper focuses on sort of a model for how those cells that are born during embryogenesis and during the post-embryonic phase, how those get connected together and start to form a coherent connectome. Because what happens in the biology is that you have neurons that are born in the embryo, and then the embryo will exhibit movements, but those movements aren't connected to the connectome. They're connected to autonomous things like muscle twitching and other cell migration things. So that's kind of what happens in the embryo. So there's no movement behavior driven by the connectome in the embryo. But what happens right around hatch is that those connections, those synaptic connections, become salient, and you start to get like goal-directed movements and more intentional movements that are driven by different circuits that we know in the adult and we've characterized in the adult.
Can I add a comment?
Yeah.
In embryogenesis, there's some cells that are loose and not really connected to each other in a proper tissue. And then they become connected and become a tissue. So they're a lot stronger and more connected mechanically when they become a tissue. And some quote unquote tissues aren't really a tissue yet because the cells are separate. They're not connected to each other. So it kind of goes across mechanical lines too, somewhat.
Yeah.
At least in embryogenesis, and I think that's probably true of homeostasis and cancer development as well.
Yeah.
I kind of divided biology into three things: disease studies, homeostasis or normal physiology, and then development. So,
well, that's an interesting division.
Yeah, well, they all seem to have similarities, but have different outcomes.
Yeah.
So, yeah, that's anyway. That's where my brain goes.
Oh yeah, I think that's interesting. Yeah, certainly, like in biology, like some people talk about homeostasis and some people don't. Like the physiologists are big on homeostasis, and but if you think about like other subfields, they don't talk about it as much. And so yeah, but it's interesting to think about, like you have homeostasis, development, disease. I guess plasticity would also fit under development.
Yes.
You'd have, like in disease, you might have a lack of plasticity, or you might have too much plasticity in the case of like cancer. And so it's like that you have these subheadings under each one. Homeostasis, how do you have plasticity under homeostasis? So yeah, interesting. So yeah,
That just it's the way the literature is
right.
on the subject.
Oh, what do you mean the literature? Like,
well, the literature seems to break up into those three categories.
Okay.
When you're looking at biology and trying to find out about the mechanics of cells, and that it seems to break up into those three topics.
Okay.
In those three areas.
So this would roughly be mechanobiology.
Yeah.
Yeah. Right, that's great. Yeah, so this second paper, this is where we have a bunch of different uh, data sets and figures. So these are a lot of CSV files, and some of these are kind of like analyses, and the chemical connections are actually secondary data sets that have been put together. So like this was from a data set which is described in the paper, um, and that was a data set which was made available, and then we took that data set and reanalyzed it, and then linked it to some of the data sets we had in the first paper, and produced these new composite data sets that describe the chemical connections or the synaptic connections between neurons in development. So this is a little bit different way of doing this because the available data sets involved information about birth times of cells and their differentiation. So like, when cells terminally differentiate, that's considered their birth time, because you get a division, you get divisions of developmental cells, and then when you get this terminal differentiation event, you usually get a final division where a developmental cell will divide into two cells that then differentiate. So that's what I mean by birth time. And then you have information about synapses, but those were measured in larval worms. So after the worm hatches, it goes through 4 larval stages, and then it reaches adulthood. And in those larval stages, there's a lot of plasticity in the synapses. And so, there are going to be a lot of changes in the synaptic weights, and some of them are transient, some of them are stable. And so we have that information. The question is there a pattern between specific types of neurons and specific times that they're born and these synaptic relationships? And so that's one question that was answered here. There's some other questions that are also answered in some of these data sets. And then there was a game-theoretic model that was applied to these data to do this analysis. So this is the part of some of these other directories have to do with that. So that's raising the worm brain, and that is linked to this paper, which is raising the connectome. And this is published in Frontiers of Cellular Neuroscience in 2020. So this was something that was done kind of during the pandemic. So this is going over these data and the model, and talking about something called connectogenesis, which is the origins of a connectome. And so this is also available online. You can read more there. Okay, so that was those two. And I wanted to highlight that because those data sets are available for training models, because they were analyzed, but they're analyzed using certain tools. You can train machine learning models for, I don't know if there's enough data to really train it well, but you could do you could generate synthetic data based on those data, and I don't know how you want to do that to try to make that useful as a training set, but it's there. All right. Hello, Morgan. How are you?
No, I'm good. Sorry, I'm driving.
Oh, it's fine. Yeah. All right, so let's move on to, I think I'll talk about this paper, a new bioRxiv preprint. This is Manuel Zimmer, has worked with the OpenWorm Foundation in the past. So they're doing this study called an intrinsic neuronal manifold underlies brain-wide hierarchical organization of behavior in C. elegans. The abstract reads, large-scale neuronal recordings in various species have revealed behavior-related activity patterns. So a lot of connectomics involves looking at the connections between neurons, and a lot of times you have to do that at a whole brain level. And then, making statements about behavior. So, we don't want to know just sometimes we want to do single cell recordings, but we'll often do those in parallel and then they boil down to maybe hopefully some behavior that we can identify, and then identify not just the cells involved, but the connections involved as well. Surprisingly, these activities are distributed across brain regions. So again, we don't just have one cell or one small group of cells involved in a behavior. Usually there are wider networks that are involved. And, there are exceptions to that, but most of the time, these are larger. We did a paper a long time ago. We reviewed a paper where, typically in C. elegans, we've defined different behavioral circuits. And one of those is the motor circuit, where you have about 7 or 8 neurons that are involved in basic directional motion, so forwards and backwards towards, say, like an environmental source like food. And this paper basically reinterpreted that as being sort of this 7 or 8 neuronal circuit being the core of the behavior, and then other parts of the connectome serving to modulate that behavior. So you'd have like this core circuit, and then you'd have these other areas that contributed to a tuning of that behavior in different ways. So this is not to say that like every activity involves every part of the brain. It's just that there's a lot more sort of regional and global connectivity that is relevant to, say, certain behaviors than we usually assume. And so this includes sensory areas, which are notorious for being kind of widely distributed and involving a lot of inputs. However, their origins and functions are incompletely understood. Using whole brain imaging and freely behaving C. elegans, we discover that such distributed brain activity is dominated by a low-dimensional manifold. So this, of course, is a mathematical tool. It's a space that you can map all this activity to. It's low-dimensional, so we can visualize it. And this is not, it's not a physical space like we might find in our models of, of cell position. This is, it's like, I wouldn't want to say it's similar to a latent space, but it's basically the sort of mathematical space that we can map all the variation of our data set to, corresponding to the major action sequence of C. elegans. The manifold originates largely from intrinsic dynamics, with minor contributions from movement-induced sensations. So this is where, you have these intrinsic dynamics, I suppose, are the interactions between cells, sort of the things that happen in these pathways, how information gets processed, how you have excitation and inhibition and things like that. And then you have these movement-induced sensations that contribute minorly to this manifold. We discover a new function of the manifold, to gate the activity of a large number of neuron classes. So again, this extended network, or this extended circuit, you might say, can be mapped down to this low-dimensional space. It can be described that way. We can look at the thing and visualize it and see kind of what it looks like, how it changes with behavior, with time. And there's this gating activity which occurs. So it's sort of this regulatory thing. This gating constrains them to selectively encode faster timescale motor patterns during specific brain states. So again, you have this behaving C. elegans. A lot of times, if you're in an experimental setting, you can take away all other inputs. You can study movement in a very sort of isolated setting. We can simulate movement with maybe 7 or 8 neurons. We can get the basic movements down. But that's not really the way C. elegans behaves, especially in the wild, where the native environment for a C. elegans, a nematode, is not an agar plate, but it's this 3-dimensional volume of soil where they're trying to move through the soil, and they're consuming bacteria. They're moving up the column and down the column, And that of course introduces a lot of different types of brain states, a lot of sensory states, a lot of other types of things going on in the brain along with movement. And so those, movement is contextual in that way. Everything is gated through this larger network. We therefore propose that one principal function of brain-wide dynamics is to enable hierarchical organization of behavior. Information about major actions is broadcast via a distributed manifold to enable nesting of granular motor patterns within those major actions. So there are 2 points here. The first is that, 3 points. The first is that they propose a hierarchical organization of behavior. You have the movement itself, you have the things that influence movement, and you have the sequences of movement that can be broken down to this hierarchical model. So that's one thing. The second thing is that they talk about a manifold, almost as being like this thing you'll find in the biology itself. And this is something, a theme you see a lot in computational neuroscience, where they talk about manifolds. And you could use a manifold to sort of describe the data, but you can also make the assumption that there's this manifold that occurs within the brain or within the connectome. And so, the question is the connectome or the cells that are being connected, do they use this kind of model, this representation, to guide the global state? Or is this something that we can extract from the data and the data fit this model, but is something else that the biology is doing? So it's an artifact of information processing, but does it really exist as a computation in the brain? And then the third thing is that we talked about an article at our Saturday morning NeuroSim meeting about brain emulation and C. elegans. And I think Morgan remembers this, what I'm talking about, which is where someone wrote an article about the different attempts to emulate or simulate the entire connectome of C. elegans and thus simulate the entire brain of C. elegans. The C. elegans is a very small brain, 302 neurons. You can have this sort of whole brain emulation at a fairly low computational cost. And so the article kind of walked through the different attempts at talking about OpenWorm as being one of the latest attempts, and almost talking about like how OpenWorm failed because there wasn't this, it didn't achieve the goals that the author of the article wanted to achieve, which is total brain emulation, whatever that means. And Morgan is familiar with some of the people involved in brain emulation. It's a whole field that exists. And the idea is that, yeah, Morgan, did you want to say something?
Well, I was just going to say that I'm driving up Northern California to a meeting of basically all those people.
Okay, yeah. So timely.
It's very timely.
Timely. And so basically the idea is that, you have simulation of the connectome. So we can simulate the connectome. We can get behaviors out of certain neurons. We could even simulate this manifold activity where we say, okay, there are other cells that modulate the movement and all this. And then there's emulation, which is something else. And, I don't know, I've read a little bit about it. And it's kind of like simulation, but also putting something in the computer model that is almost like sentience or consciousness. It's not really clear to me. Like that's my impression of it. But whatever it is, it's emulation is like maybe a step above simulation. And so, like, I think it's interesting in light of this article because you have this sort of model. We have like a lot of good data that's coming out all the time regarding the connectome. The connectomics in the last 10 years has made a lot of strides, especially in C. elegans, where you have these data sets that are specific to males, specific to hermaphrodites, specific to maybe to mutants. and we have a lot more data per neuron. So we have not just the information about ion channels and other things, but we're increasingly getting information about real-time behavior and the context of movement. So we have these circuits, but the circuits generate the movements under different conditions that the worm finds itself in. So different sensory inputs, different other types of states like aging and development and whatever. So it's really interesting. So that's the summary of the article. They kind of talk about some of these connectomic studies. So recent studies have discovered neural activity correlates of instantaneous behavior distributed throughout the brain and surprisingly include sensory areas, or I guess surprisingly including sensory areas. These observations were made across animal phyla from invertebrates to mammals, including primate cortical areas outside motor cortex, although to a significantly lesser extent. The origins and functions of these activity patterns are not known. That's reference 9, which reference 9 comes up as this paper Kaplan and Zimmer, brain-wide representations of ongoing behavior, a universal principle. So this is, I guess, these dynamic representations, and if these apply to all sorts of animals with brains. So Manuel Zimmer is a pretty interesting person. He does a lot of really good work. Neuronal population activity patterns within specific brain regions, however, are well described. So we don't necessarily know about some of these sort of activity patterns in connectomics. We can't make a lot of statements about what they do and what, why maybe they are related to the movement under study or whatever, the behavior under study. But we know about neuronal population activity patterns within specific brain regions. So we have these population studies of neurons. So there's, I guess, a gap there. Such activity patterns are often modeled as smooth trajectories in a low-dimensional neuronal space, termed a manifold. So in primate motor cortical areas, both the planning and execution of arm reaches have been functionally linked to neuronal trajectories on low-dimensional manifolds, demonstrating that manifolds can encode relevant aspects of behavior. So yeah, this is a classic area of this sort of modeling and recording. So in primate motor areas, you usually have like premotor cortex, motor cortex. You usually have a population of neurons that are responsible for directional movement of arm movement. So you have usually, they'll do an experiment where a primate will be sitting in a chair and have to reach for a juice box, and that, they'll put it in different locations. So the reaching is in different directions, and then they record from a population of neurons, and that population of neurons are doing things that are somewhat stochastic. They're firing, and sometimes they become synchronized to a certain phase angle, which then translates into movement in a certain direction. And a lot of when they record the data, they'll do like an average of the activity, and they'll be able to replicate that movement direction. So they can actually get the recording. They can, go through the gross activity of the population, find the average, and predict the direction of movement. And they use that kind of signal in brain-computer interfaces and brain-machine interfaces. They can take the signal, do the signal processing, and then put it into a robot or a mechanical arm and produce movements in those directions. So that's actually quite useful for neuroprosthetics as well as understanding this manifold. However, it is not known whether any manifold structure underlines the assembly of individual movements into long multi-action sequences. Moreover, it is unclear how neuronal activity underlying granular behaviors, such as grasping, is nested within activity related to course behaviors, such as arm reach. So it's like this sort of, when we talked about that monkey example or primate example, there's the arm and then there's the hand. The hand does grasping and then the arm moves. So you can tell, from the data that we just recorded, what direction the arm is moving. But then the grasping is a separate behavior and that involves separate neurons. And then we want to know how all of that is coordinated together in neural activity in the connectome. So it's very hard to know. We might be able to decode, say, like one component of the behavior, but the other components of the behavior are separate and apart from that. But they're coordinated, they're synchronized. So how does that happen? So we therefore hypothesize that one function of distributed brain-wide activity is to organize patterns of behavior across these different scales. In natural contexts, animals must simultaneously perform many tasks, from processing sensory information to coordinating actions and motor patterns, all while maintaining longer-term behavioral goals. So they have to coordinate all these things in the same sort of sequence of behaviors and the same sequence of sort of being aware of what's going on. So like, in C. elegans, I don't know how much awareness there is, but, they have to coordinate a sequence of movements. So sometimes they have to go a long way to find, some sort of food source or some sort of chemical source. Sometimes they have to remember locations and they have to coordinate all of that into their movements. So it doesn't just involve a motor circuit that tells the worm whether to go forwards or backwards. There's a lot more to it. And so we can turn to ethological studies, which are the studies of animal behavior in the wild, that propose this is achieved through a hierarchical organization of behavior, in which slower timescale behaviors hierarchically orchestrate faster timescale behaviors. So if you have faster timescale behaviors, like if C. elegans wants to make a turn or wants to reverse its direction, that's a fast timescale behavior, but that's orchestrated by slower timescale behaviors, meaning that there's a goal maybe of finding a food source or a chemical source, and that goal is subsumed by these different components, these movements. And so, it doesn't just happen in a vacuum, and thus we should expect the connectome to have a connectivity that reflects that. They shouldn't just be like the movement circuit operating in isolation. It has to have more inputs, and it has to do this in a way that's structured, because it's not just adding in stuff, adding in activity. It's adding in specific information. Some studies have identified neural activity correlates of hierarchical behavioral organization in specific neurons or brain regions. However, how such hierarchical organization is achieved across the brain and the role of neural manifolds in such organization, if any, is unknown. So, again, this sort of real-time behavior in, say, like primates, movement changes the visual landscape in the opposite direction of the movement. This is termed optical flow. So if you're walking down a hallway, your view is changing because you're moving, and that movement is kind of changing the kinds of things you're interacting with. This is something called optical flow. They use it in engineering. They use it in sort of perceptual control theory and in ecological psychology as an organizing principle, because it is important to have this model of sensory inputs not as these discrete moments, but as this sort of continuous process that actually changes how the organism interacts with the environment. In many instances, such self-generation sensory information is suppressed in the nervous system. So this is not always useful, but it sometimes is useful. Multiple species use it to adjust their movements. So for example, when birds try to make landing moves in flying, or insects try to make landing moves in flying, and sometimes mammals will use it for navigation, including humans. Additional sensory modalities may also exhibit self-generated signals. For example, when nematodes crawl backwards, they sense traces of O2 and CO2 gases caused by their own metabolism. That's an interesting example. Overall, such sensory-driven activity is highly correlated to the animal's behavior and may be prevalent in the nervous system. Thus, disentangling the origins of brain-wide behavior-related activity is required for a comprehensive understanding of its functions. So let's go down to the figures. I think those are probably going to be interesting. So what they do in this paper is they build this pipeline to record and quantify whole brain activity and behavior in C. elegans. So they're doing this whole brain neuronal calcium fluorescence imaging in freely crawling C. elegans. So they're actually recording it. I guess the C. elegans are in a dish on an agar surface, and they're crawling around and they're getting the information that way. We therefore designed an assay arena where worms freely explore bare agar pads under a cover glass. So they actually have this under a microscope. They have the agar pad here, and they have a cover glass on top. Brain activity was imaged with high magnification immersion optics, and behavior with infrared illumination and low magnification to capture the entire body of single worms. So that's how what they recorded. They also had transgenic worms, and they wanted to use that to look at different mutants. To overcome the challenge of tracking the small and densely packed neurons in the deforming tissue of crawling worms, we took a segment and track strategy. So, of course, they had to correct for movement artifact by virtue of the worms moving around. They didn't have, a static worm to record from. So they had to make corrections. And they did that, I think, in Nepari. We identified a subset of neuronal identities based on their anatomy, position, and activity. Some remaining ambiguities were resolved using the NeuroPal approach for non-stochastic and unique labeling of neuronal identities. So there's this package NeuroPal, or this approach called NeuroPal that you can use to label neuronal identities. In total, we identified 61 neurons, which, after combining left and right pairs, correspond to 38 neural classes. So if you look at the names, the nomenclature of the adult neurons, you'll see that oftentimes you have left neurons and right neurons of the same name. So like ADL is a neuronal pair, or a neuronal class in this case. You have ADLL and ADLR. It's left and right. So there's a symmetry between left and right. And so this is the way the C. elegans connectomes is organized and named. And so, each side has a neuron that basically does a similar thing. And, you can compare those two to see what the difference is in sort of bilateral symmetry. And oftentimes there are differences, not great differences, but, you can imagine that there are differences in information processing in the connectome that are split down the midline, and then there are differences on the left and right side. But then, of course, so we can make statements about the left side and the right side that way. In a single data set, a subset of these were identified, representing around 40% of tracked neurons in freely moving data sets, 38% of tracked neurons in immobilized data sets, and around 15% freely moving GFP data sets, which is different than CGAMP. And, it's just the different ways of recording the information. So this is a figure of kind of what they were doing. They had these recordings of neurons. They were able to find PCA modes down here, and then they were able to correlate that with the speed. So they actually had, I think in A, they had a heat map of neuronal activity. So this is 121 tracked neurons across time. And so these are each neuron going down the y-axis. time is from 0 to, I guess, 500 seconds. And you have these behaviors that were identified. You had these PCA modes, which are like breaking down the behavior, and then these neurons, which had an activity. And then they looked at the PCA-CCA similarity. So that is where we have neuronal only is PCA. So this PCA is actually from the neuronal activity, and then that was mapped to the behavioral activity or the speed. And then continuous behavioral neuronal, which is CCA, and discrete behavioral neuronal is CCA discrete. So we have this, once we collect the data from both sources, we can like look at the PCA analysis of neuronal activity and the CCA analysis of this behavioral and neuronal activity. So that's what these graphs kind of represent. And then this graph here, C and D, which show kind of the correlation between PCA and CCA and for different PCA components or PCA modes. So you have 1 through 3 here, and then you have PCA mode 1, 2, 3. So it looks like PCA mode 1, it has a high degree of PCA-CCA similarity, and then the rest of them less so. And that's, I guess, kind of true PCA because the first component is always the strongest, although I don't know what they mean by modes here. The first component usually has most of the variation. In any case, so let's move down to C through G, or E through G. So these graphs represent neuronal components, behavior and neuronal components, and discrete behavioral neuronal components. So this is the PCA on the left, the CCA in the middle, and the CCA discrete on the right. And you have PCA versus PCA, and I guess this describes the amount of variation. And then this is this dynamical trace of the activity. So this shows sort of what those look like. Then you have CCA weight for different neurons. So you could isolate this down to different neuronal classes. So it's like the left and the right. So AVA left and AVA right, AV left and AVA right, and so forth. And so you could take the CCA weight that you're getting from the behavior and the neuronal activity, mapping it to these specific neurons, and then you get this AVA being, I guess, the highest CCA weight, and you go down to negative values down in these neurons here. So they actually have this broken down by neuronal classes. And so this is figure 2. This shows the difference between freely moving and immobilized worms. So you have freely moving and immobilized contexts where you're recording. So again, this just shows the difference that this sort of free moving paradigm makes. Freely moving, of course, gives you different activity than immobilized. And again, we have this map of PC1 weight by neuronal class. So we have AVA and AVE at sort of the top of the highest weights, and then you have negative weights down here for this host of neurons. So again, we have this for immobilized and freely moving, and so there are differences in some cases between immobilized and freely moving, and in some cases less so. Then there's this change in dimensionality in freely moving. So just looking at freely moving, you have this change in dimensionality by neuronal type. So you have sensory neurons, interneurons, motor neurons, interforward and interreverse, meaning that they're involved in forward and reverse movements. They're interneurons, but they're involved in these specific behaviors. What you see here is you have, well, quite a smattering of different outputs here. You have the motor, so you have motor neurons that are kind of spread across this in terms of their log p-value. And then you have, of course, the change in dimensionality, negative and positive. And so this is just showing for these different, I don't see any real pattern here. Interneurons are interspersed across this graph. So are the forward and the reverse. Actually, I think the forward might be all positive and the reverse are tending towards negative, but I'm not really sure how to interpret this graph. In any case, I think there are a lot of graphs here, probably if you go through the paper more. This is another interesting graph here. This is in figure 3. This is figure 3, I want to say J, or I, I'm sorry. This is figure 3I. Let me zoom in a little bit here. And so we have, let me get this. Okay. So this is freely moving GCAMP versus free moving GFP. And so you can't really tell the colors. I guess one is gray and one is purple. But this is hierarchy model performance. So I guess this is fitting a hierarchical model of behavior. And this is behavior only model performance. So this is comparing these two different types of model performance. And then for these different, from these different data sets, we have for specific cells, these results. So it looks like you have some neurons that fit neither model, and other neurons that fit both models somewhat well. So again, you have the cells that are labeled, and the differences between freely moving for GFP and GCAMP, and those are just different ways of measuring it. But basically, you have this trend towards, I guess, hierarchy model performance. There's more variation there. But it's interesting, again, to see which cells perform differently for different types of model performance. And that may tell us maybe, which cells might be involved in a behavioral hierarchy versus ones that are just maybe more core to this behavior. I don't really know how else to interpret it. But, this letter K, which is, of course, another part of figure 3, this shows a radial graph of, I guess, looking at different points in the worm, different points in the behavior. So the worm is moving, and it's making this undulating movement. So different parts of the body are responding differently to the movement. And so then this is behavioral only model performance. So this is looking at, I guess, how the behavior works in the model. This is like the direction of movement, and then these are the cells that are involved in this. And I guess these are classified by neuron type. So we have dorsal motor neurons, we have interneurons, we have ventral motor neurons, and they're implicated in different angles of movement. So we have, of course, these different points of, so as the worm is undulating, they're moving in different directions, different parts of the body. And so you can see that there are different neurons involved in those different components of the movement. And then, of course, there's this turning mode measure, and then different specific neurons here, and then their class. So they actually show how for turning mode, you have some neurons that have a wide amount of variation, some with a very tight amount of variation, and then some are sort of in negative turning mode and some are in positive turning mode. So this is, and of course, I think most of the motor ventral neurons are involved in the positive turning mode, and most of the dorsal motor neurons are involved in the negative turning mode, as I think we see here. Breaking that out in a different way. Okay. So yeah, so that's pretty much this paper. And this is kind of summarizing. We observe rich neuronal dynamics encompassing a large fraction, around 80% of all tracked neurons. A striking feature of these neuronal population dynamics are correlations among many sensory enter in motor neuron classes. For most neuronal cell types, this correlational structure, quantified by PC weights, which is the principal component analysis we showed earlier, was reproducible in immobilized conditions. However, both conditions showed frequent transitions in these correlated activity patterns. Importantly, in the immobilized condition, animals were not only deprived of external acute stimuli, but also lack of any time-varying afferent inputs or proprioception of their own movements. So they didn't get this feedback that they might need from the environment to sort of control their behaviors. It's all feed forward, which is interesting in the immobilized condition, which is interesting because there's often this, the celebrated role of feedback in sensory motor control, and that it's such a critical source of sort of control, meaning that, if you're moving towards a goal, you have to get these corrections constantly, or else you can't really make it to your goal. You don't get that kind of refined set of movements you need. This result supports the idea that these dynamics are intrinsically driven. So it may be that you get intrinsic processing instead of constant feedback from the environment. This does not exclude that the environment can influence these dynamics and their associated behaviors. For example, we've shown that constant or only slowly changing arousing factors such as time off food, hunger, or experimental conditions such as excitation light can affect the frequency of behavior or brain state transitions. So that's interesting that you have these intrinsic dynamics that are providing this feedback to the goal, and it's maybe only augmented by things from the environment. And that's, the assumption is that the environment is really kind of providing every single bit of that feedback. However, such factors cannot explain exact transition timing in the observed correlational structure neuronal dynamics. So that's good stuff. And, this really kind of gives us a view as to this diverse continuum of behavioral representations in the C. elegans brain, of course, the adult brain, and there are a number of implications for sensory processing and really understanding how that works. C. elegans has a wide variety of sensory processing, largely chemical processing, and so this is, it does a lot of really interesting behaviors with respect to chemical processing. They can follow plumes, they can follow gradients of different chemicals. So they have some really complex behaviors which we might not have thought about. And of course, this reflects their natural environment, right? They're in this three-dimensional matrix of soil. There are all sorts of stimuli around them. There's rotting fruit, there's bacteria in different places, or different types of toxins. And the idea is they want to be able to stay alive and consume food and move around in that column of soil and reproduce. That's their goal. And so, there are a lot of things that they need to do in terms of processing chemical information, plume following, things like that. So that's all kind of what this has to do. There's a lot of navigation and a lot of sort of multidimensional sensory processing. Okay, so I think that's all for that paper, and that's pretty much all for today. Did we have any comments before we go? Anything you wanted to mention? Go ahead, Susan.
Oh, nothing. Just thank you for the presentation.
All right.
I'm interested in neurons, so.
Yeah. All right. All right. Talk to you next week. Thanks.
Okay, bye.
Bye.